ground. So you would have definitely come across this word enzymes. So guys, enzymes, if you see, they are nothing but a protein that helps to fasten the reaction process. They act as a catalyst. So if you see here, as per the scientific definition, an enzyme is a protein that act as a biological catalyst. Means it is obtained from living organisms, be it plants, be it animals, or be it microbes. So their main function is to speed up the rate of reaction, that is speed up the rate of chemical reaction. So they act as a catalyst, they enhance the speed. For example, if normally if to bake a food, it might take you said 10 I mean 5 hours, but if you add particular enzyme, so it might reduce the rate of baking or cooking the food by almost half. So this very basic example would give you the idea that how these foods sorry how these enzymes, they fasten the process, right? They act as a catalyst. So now moving ahead, there's another third important, very important point. The enzyme is not destroyed during the reaction, and it is used over and over. So this is a very basic example. If you see curd, so everyone would have consumed curd. So lactobacillus bacteria is present in that, right? So those bacteria, so basically they give that texture to the food, and basically how they does is, that is that is the enzymes that are working right in that. So and also you might know that if you take curd and if you next day you add fresh milk to it, so that milk is also converted into curd. So it means these enzymes or the bacteria which is present in the food, it never dies, it can be carried over and over again, provided they are given the food to eat, like milk, lactose, and the milk is the food for those microbes. So until and unless they are getting their favorite food, in this case, in the case of curd, it's the milk. So so as and when they are getting the food, they are happy with that. And the sugars that is present in the milk, so they break down those sugars, they convert it into curd. So this is a very basic example which I would like to give you so that things are clear with you. So now moving ahead, why microbial enzymes? So why microbial enzymes are the preferred method? As I said, enzymes, there are other methods as well to fasten the reactions, but why enzymes only? Why are they preferred? So if you see, microorganisms has been used in food fermentation since ancient times. So not only now, since the discovery of the food by humans, so these enzymes has been are being used, right? Just a second, guys. Is my voice audible? Can you please confirm once? Is it audible, guys? Okay. So if anyone is having issue because someone has messaged me that your voice is not audible, so please just log out from the meeting and please log in again. It can be an internet issue at your end, because everyone can listen it. I got the message from the students, so they are like voice is audible and clear. So if anyone is facing any technical issue, not to worry, just log out from this meeting and log in again. And in case you miss any part, I would be sharing the recordings, so you can cover from that. Not an issue, right? So guys, so I was saying why microbes are the preferred method, because if you see the second point, they play a major role in food industry because they are more stable than plant and animal enzymes. So as I already said, enzymes can be obtained from animal sources or plant sources, but the enzymes obtained from the microbes, these are the more stable than any of these enzymes. Because thing is, like microbes, as and when they get their food, microbes can grow anywhere. Even on your body there are millions and billions of microbes. Microbes are everywhere. But the for example, if you're taking enzymes, especially if you're taking enzyme from the plant or the animal, there is a lot of complex processes that you need to go. But microbes are such an organism which are present everywhere, be it your body, be it your food, be it your study table, be it your mobile. be it your laptop, everywhere. I mean, everywhere. They are easily found. So that is why these are one of the preferred, these are one of the preferred microbes. I mean, one of the preferred method which could be used in food for fastening the reaction, and that this fact was really understood very fast by food industry. So that is why they are leveraging these enzymes to enhance the texture, to give the flavor to the food, to break down the fats in the food. So we'll discuss all this in detail in coming slides. So not only this, if you see the third point, enzymes are also cost effective method with less time and space requirement. Definitely, guys. I mean, if and as I assume, since you all are science students, so you definitely, you would have worked in your lab with microbes, right? You would have done microbial plating and all that stuff. So you can see, I mean, in a small space, billions of microbes can grow, right? In your petri dish is just a small plate. So in that, billions of microbes can grow. So that is why these are very cost effective method, and they require very less time and space. So that is why they are one of the preferred method. And fourth is high consistency. High consistency means these microbes can be used over and over again. You need not to grow them again, you need not to culture them again, you need not to incubate them again. You can just take one loop of culture and you can use it again and again over a period of time, until and unless provided they are given their favorite food to eat, right? I hope it's clear, guys. Just give me thumbs up if everything is clear till here. Okay, fine. Moving ahead. So now, guys, as I mentioned, so since we are going to learn about enzymes in this, so this is the summary basically. So we'll be covering 8 important enzymes, right? So we'll be covering 8 important enzymes in this slide. So there are even a large number of enzymes which are used in food industry, but I have taken the 8 important which are majorly used in food industry, especially for the processed foods, right? Yes, microbes produce enzymes, guys. Someone has asked me the question how enzymes are produced. Microbes, they eat the food and they produce the enzymes, right? So we'll be going through alpha amylase, protease, lactase, esterase, lipase, cellulase, pectinase, and glucose oxidase. So moving to the next slide. So let's first talk about alpha amylases. So alpha amylases, if you see, I have put relevant pictures over here. So majorly these enzymes are used in baking industry or brewing industry. I mean, for production of beer, sugar, right? And wine industry. So yeah, brewing industry and baking industry, they are majorly used. So alpha amylases, they are majorly produced by this bacteria. Everyone would have heard about this, the name of this bacteria, that is Bacillus subtilis. So we'll learn about this in detail. So moving ahead to the next slide. Alpha amylases. So first, let's understand what these enzymes are and how do they, carry out their activity. What do they attack? So if you see here, alpha amylases are starch degrading enzymes. So if as and when you hear about the amylases means they would definitely be attacking on the sugar compound in the food. So if you see here, they attack the starch, they are known as starch degrading enzyme, and they have the capability of hydrolyzing alpha 1 for glycosidic bonds. So this is the major bond that they break down, right, in the food. So these requires calcium ion for their activity. So they require calcium ions. Now, guys, try to relate it as we read in our first lecture. So calcium, these are the important minerals which fall under the category of micronutrients, right? So and yesterday also we learned that calciums, there are certain anti-nutritional factors which are present in food and they can bind with calcium. For example, oxalates, they find with calcium and they become calcium oxalate, and it reduces the volume of the calcium in our body. So if you see alpha amylases, they require calcium ion for their activity. So these enzymes are, just a second. So guys, is my voice audible? Because again, few students are saying, sir, your voice is not audible. Can you please confirm? Okay, so guys, now if anyone is saying your voice is not audible, I am not going to take that. Please, if my voice is not audible, check the internet at your end, please. Right? So coming to the third point, they are widely used in baking industry. So baking industry, for example, cake making and bread making, then waffles, all that stuff that comes under bakery. So they are majorly used in baking industry for flavor enhancement and anti-stalling agent to improve bread quality. So they act as a flavor enhancers and they improve the quality of the bread. And how they does is they just break down these complex sugars to the smaller sugar molecules, and those helps these enzymes to give flavor and improve the bread quality. So if you see the application, as I already mentioned on the first slide, they are majorly used for baking and brewing. And if you see the baking, in baking, alpha amylases are added to the dough for the conversion of starch into smaller dextrins. So they convert the sugar, complex sugar, that is starch, which is present in the dough, in the flour, atta. So it breaks down the sugar into smaller particles, that is known as dextrins. So now, also you might be knowing yeast, Saccharomyces cerevisiae. This is the one of the major, it's yeast that is used in baking industry. So that yeast now it can ferment these dextrins, and it improves the taste, crust, and color and toasting quality of the bread. So this is the major reason why alpha amylase is used in the baking industry, because alpha amylase is on the first stage, it breaks down the complex sugar into simple sugar, right? And then that simple sugar is used by the yeast, right? It is used by the yeast to increase the taste, color, and toast and quality of the food. Then coming to the brewing. So in brewing, that is in ethanol production, starch is converted to the fermentable sugar. So if you see here, amylase function is nothing but to break down the complex sugar into simple sugar. So it hydrolyzes, it breaks down this glycosidic bond, and it converts the starch, that is complex sugar, into simple sugar, so that further yeast can further act on that and convert it into the end product. So if you see here, further fermentation of the sugar to the alcohol is carried by Saccharomyces cerevisiae. So again here, alpha amylases are further helping the yeast to reach out the end product. Right? I hope it's clear. So now moving to the next slide. So now we'll talk about proteases. So proteases, again, if you see, they are majorly used in baking industry and dairy industry. And the microbes that they are obtained from is Bacillus thuringiensis. So guys, so in this lecture, in this topic, what you need to remember basically is the name of these enzymes, major enzymes, and the bacteria from which they are produced, and the industry in which their application is. So that is the only important thing you need to remember. Right? That's it. Moving ahead. So if you see proteases, so proteases are the enzymes which catalyze the hydrolysis of peptide bond present in the proteins. So from the name itself, it's clear, protease. I mean, something it has to do with protein. So these enzymes, they again further hydrolyze. I mean, they break down the peptide bonds, since protein are the complex, like they are long chain. So they further break down these peptides into simple proteins, right? Smaller chains, and they represent 60% of the industrial enzyme in the market. So guys, you can imagine from here the amount of application, they are widely used in food industry, proteases, how it has 60% of industrial enzyme in the market share. So in the market, 60% of enzymes that are used in food is protease enzymes. And these enzymes, plant proteases such as bromelain, ficin, papain, are widely used in the food industry. So these are the three major enzymes. These are uh, uh, are the major enzymes which are used in the food industry, and these enzymes are obtained from plant basically, right? So if you come to the application again, baking and dairy are the major, but also if you see here, they are widely used in brewing industry, tenderization of meat, and coagulation of milk as a digestive aid, and they improve the flavor, nutrition values, solubility, digestibility of the food protein. And how it does is, that is not important. You just need to remember the enzyme and the microbes from where they are produced and the industry in which they are used, right? Just for your knowledge sake, you can remember these, how it does if you require, but from examination point of view, that is not at all important, right? So coming to the baking industry, so baking, as you all know, so they would be used in bread and baked foods, crackers, waffles. So everyone would have eaten these, right? If you visit any supermarket, then definitely you can see breads, crackers, waffles. All these are available in the market, and if you see the label behind, so you can see these protease enzymes being mentioned if you are buying any processed food. So further, what they does is they reduce the mixing time, decrease dough consistency, uniformity, and regulate the gluten strength in the bread and improve the texture and the flavor. So majorly, it makes the dough ready for baking. If you see here, it decreases the dough consistency and uniformity, and it further regulates the gluten. It strengthens the gluten which is present in the bread, so that it can be baked properly, and it gives the proper texture and flavor. So whenever you go to the supermarket now, there are variety of breads which are available in the market. So and the major thing is why they are being used, because of the wide application that they have, right? I have already mentioned. So coming to dairy, so now they are also being widely used in dairy industry. Why? For cheese ripening, to modify the functional property and reduce allergenic property of the milk products. So proteases, as you all know, they break down the complex proteins into simple compounds. So here they help in cheese ripening, right? I mean, since normally the cheese ripening might take a long time, but in industries they add some proteases, they add some enzymes that fasten the process, because industry has to serve large number of customers and they want to make profits. So to achieve all these factors, so industries use these methods. They use enzymes, natural techniques to fasten the process and make the profits, right? So now coming to next enzyme, that is lactase. So again, if you see lactase, it's majorly used in dairy industry, especially in ice creams, and also again in dairy industry for cheese. So lactase is enzyme which is obtained from again Bacillus subtilis. So now moving ahead, if you see lactase, these enzymes, they catalyze the hydrolysis of lactose. So lactose, all you know, lactose is a milk sugar, and the name also, from the name also you can recognize it's a lactase. Lactase enzyme, definitely it has something to do with lactose, and lactose is a milk sugar, and it belongs to the family of hydrolysis, and it can be obtained from numerous biological systems including plant, animals, and microorganisms, right? So if you see these lactases, they can be obtained from multiple sources, but enzyme is the microorganisms are the preferred source. And why these are preferred source, we have already spoken about this in our first slide. And further, bacteria, fungi, and yeast is preferred choice due to higher yield and relatively low cost. So that is what I mentioned in my first slide as well. Bacteria, yeast, and fungi are the major source to obtain these lactase enzyme. Why? Because they give higher yield, they require less space, they are highly consistent. I mean, even a single inoculum from the your petri plate can give rise to million of microbes in your food. So that is why they are highly consistent, they are highly useful, right? So now coming to the application. So application, if you see, lactase is used with milk and milk-based product to reduce lactose intolerance in people. So since it acts on, it for lactase also break down the lactose sugar. So lactose, it's one of the major cause of the lactose intolerant. I was reading one survey. So almost, more than 80%, 70 to 80% people in India are especially lactose intolerant. Lactose is nothing but sugar which is present in the milk. So if these many people are lactose intolerant, so now what to do? So now what industries are doing is they are using this lactase enzyme, so which can be used in milk and milk product to reduce lactose intolerance in people. So this enzymes break down the lactose sugar, so then whoever is lactose intolerant can consume those dairy products. And also if you see here, the scoopability and the creaminess of the ice cream improves significantly after hydrolysis of lactose with lactase. So definitely you would have eaten, ice creams, so those beautiful scoops if you see here. So these scoops, have you imagined how these beautiful scoops they remain in this shape whenever you take out, right? So this scoopability, the way they are taken out, they remain in this shape because of the lactase enzyme. So this enzyme ensures that these ice creams have they have proper shape. It gives them proper shape because of this enzyme after the hydrolysis of lactose with lactase. So now further in whey. So guys, whey is basically nothing when you make cheese. So whey is the byproduct of the cheese. So this whey, if you see, it's very, very high in COD and BOD. So COD and BOD are the factors which are very, very harmful for our environment. Although this is not important from the examination point of view, but I just wanted to give you the example how whey is, creating environment pollution because they are rich in COD and BOD. So this COD and BOD, this wastewater from the whey industry, when it is released to the streams, unfunctional streams, so that further leads to the environmental pollution. So this is just for your knowledge. You can just ignore this, nothing else. I just wanted to give you this point. So if you see, whey is a byproduct of cheese production, and its main components are lactose, protein, and minerals, and this causes critical environmental issue associated with the dairy industry. So in dairy industry, lactose further helps to further, if this since it's used for cheese making, so this lactose further breaks down this complex proteins in further, into the smaller molecules. So that is why it reduces the, COD and BOD levels in the waste stream that is released. Otherwise, if directly the waste from the whey industry especially is released, so it would lead to high environmental pressure. So now coming to lipase. So lipase, as the word suggests, it has something to do with fat, lipids, right? So these lipase, they are obtained from Pseudomonas aeruginosa. So again, if you see from the picture, this is majorly used in baking industry and wine industry. So as the name suggests, it has something to do with your lipids. So let's see what its major function is. So if you see here, lipases, so these are the enzymes which catalyze the hydrolysis of long chain triglycerides. So you all know, in from our first lecture we have read about triglycerides, right? So triglycerides are the we read in that presentation in our first class, if you remember. So triglyceride compose 85% of the fats which are found in our food. So these lipases, these enzymes, they catalyze the hydrolysis of the long chain triglycerides. So what they do is they break down these triglycerides, I mean complex fats, into these smaller molecules. So basically, if you have seen till here, so these enzymes, they do nothing but they just break down the complex chains into simple molecules so that it can be further degraded, right? So again, lipase also, it is doing nothing but it is just hydrolyzing or just breaking down the triglycerides, fats, complex fats into simple compounds, and they are naturally present in stomach and pancreas of the humans and other animals, especially in digestion of fats and lipids. As you all know, these lipases are also present in our body. What they do, so whenever we consume fats, whenever we consume food, I mean, Indian food, it's oily basically. Majority of our foods, especially during festivals, be it samosas, be it our sweets, or be it anything. I mean, from north to south, east to west, we use a lot of oil in our food, right? So these that oil which goes in our body. So lipases ensure that they further break down those fats so that we can easily digest them. So now if you see, the application of the lipases is majorly in food, then biofuel, even in detergent, even in animal feed, leather industry, textile industry, paper processing. So these are majorly, yeah, I mean, lipase have wide application. So how wide application? Where and ever there is a fat associated. If you see leather industry, in leather industry, majorly what is leather? Leather is made up of animal fat or animal skin. So that is where lipase is used. Lipase basically break down those fats to give the fat, to give the skin of the animal a proper shape, right? So leather, it's used widely in leather industry, and further it's used in detergent industry, biofuel, animal feed, paper processing, everywhere. So if you see here, in the food and beverage industry, lipases first find major application in dairy, baking, fruit juice, beer, wine industry. So in the food industry, they are majorly used in all these. I mean, they are used in milk. So milk has fats, even in baking, even in fruit juices, beer, wine industry, everywhere. It's present everywhere. So now if you see, different type of cheese can be made by using lipases from various sources. Yeah. I mean, whenever you go to supermarket, you would have seen, I mean, variety of cheese available in India right now, right? I mean, be it Romano cheese, be it, cheddar cheese, or be it any cheese. I mean, thousands of varieties are available. There was a time when only homemade cheese was available, which was made at home by our mothers. But now when you go to the market, there are a variety of cheese. Have you imagined how those cheese are made? They are made from different variety of lipases. Now lipases also have different varieties. If you see here, Romano cheese, so this is used from lamb of pre-gastric lipase. So this is the animal source. So majorly in India, this is not preferred because, in India, majority of the population is a vegetarian population. So Romano cheese, basically these are, from European countries cheese, Switzerland, most majorly the European nations, they are, founder, and they are the pioneer in cheese making. So this Romano cheese, this is majorly from Europe basically. And the lipase enzyme which is procured, that is procured from the lamb, from the lamb. I mean, everyone knows what lamb is, right? So from there, pre-gastric juice, from that lipase is obtained and Romano cheese is made. Now then, coming to the Camembert cheese, it uses lipase from Penicillin camemberti. So the another variety of cheese, that is Camembert cheese, for that the lipase is obtained from Penicillin camemberti. And if you see the cheddar cheese, so cheddar cheese is very, very common. I mean, you go to supermarket, you go to dairy section, cheddar cheese is everywhere in India now. So cheddar cheese, it uses Aspergillus niger or A. oryzae. So guys, by the way, these are the name of fungi. If you see Aspergillus niger, this is a fungi. A. oryzae, it's a fungi. So these lipases are obtained from fungi, right? So lipases catalyze could improve the texture and softness of the cheese. So if you see cheese, basically, cheese is very soft, it's very tender, and its texture is smooth. So that is what, how, we assess the quality of the food. So in case of cheese, we check how soft it is, how tender it is, how fruity it is, how tasty it is. So all these factors, these are given by all these enzymes, right? And coming to the second application, it is also used in alcoholic beverages like wine. The aroma can be modified using lipases. If any one of you had wine, since wine is really good for your heart, right? So wine, the taste of that wine, the smell of that wine, the aroma, that, fruity smell that comes from the wine whenever you open that bottle of fresh wine, so that is majorly because of lipase. So in wine industry, so the texture and aroma can be varied. It can vary significantly depending on the modified, it can be modified using different kind of lipases. I hope it's clear till now, guys. Just give me thumbs up if things are clear. Okay. Fine. Thank you guys. Thank you for confirming. Okay. So now moving ahead. So now let's talk about cellulases. So cellulases, if you see, it has something to do with cellulose, right? So they are majorly used in juice industry and ketchup industry. So if you see cellulases, these are obtained from the microorganism that is known as Pseudomonas fluorescens. So just remember the enzyme and the microbes from which they are obtained and the industry where they are majorly used. And also guys, whenever you go to supermarket now, so just pick up those packets and start reading the ingredients. So that is how you prepare for this exam. It's not you just read on the internet or you watch any video. Now start practicing it in your everyday life. Go to your kitchen. First start from a kitchen. So whatever processed food you get, be it anything, I mean, be it your sugar, be it your salt, I mean, biscuits, each and everything, even your milk packets. So start reading what is mentioned on that. So that is what is going to help you in the exam. Because if on any point if you get stuck, you would easily relate it. I mean, if something comes with lactose is a milk sugar. So if you read the ingredients which are given on the milk packet, I mean, the tetra packs which are used, um, I mean, there's a lot of information given on tetra packs, especially if you take that daily package, I mean polythene one, that it does not have much information, but the tetra packs have a lot of information. So guys, just keep, make it a habit. Whenever you see any food product, just go and grab it and see, read it importantly. It's very important to read. That is what is going to prepare for you the exam. So moving ahead, so cellulases. So what are cellulases? These are the enzymes that act on the polymeric cellulase and hydrolyze beta 1 for linkage to replete glucose unit. So now these cellulases, they act on the beta 1 for linkages to liberate the glucose molecule. Glucose, as you know, it's a simple sugar, right? So cellulases, they act on the polymeric celluloses, I mean complex celluloses, and they break down the beta 1 for linkage and release this simple glucose molecule so that it can be used easily, right? So celluloses from fungi, that is Aspergillus, Trichoderma, and bacteria, Bacillus, right, are potentially used in the production of the food. So these are the another sources. I mean, not only the bacteria, but also they can be obtained from fungi. And the most common fungi is which from which it can be obtained is Aspergillus and Trichoderma. Now coming to the application. So if you see the application, it's in juice industry, cellulases are applied in combination with other macerating enzymes for increasing process performance and yield and improving the extraction method, clarification and stabilization of juices. So guys, definitely I am sure you might have drank real juice, right? Real juice, then Tropicana, then Paper Boat. I mean, there are thousands of brands which are available in the market. So have you ever imagined how these guys, I mean the industry, how they clarify these products, how they stabilize these juices, which can be stored in the package for like 180 days, almost 6 months. So these are the enzymes along with the packaging. Packaging also plays a major important role in the food package preservation and transportation. So we'll talk about this in packaging section when we talk about the packaging. That's a separate topic altogether. We'll also cover that in detail. But apart from that, how food is, how it's given the texture, aroma, that everything comes from these kind of enzymes. So these cellulases are majorly used in juice industry. So now whenever you go to the market, when you see the real juice or Tropicana juice or Paper Boat, just pick those packet and definitely you will see one of these enzymes being used, right? So it also reduces the viscosity of the nectar and puree from the food such as apricot, plum, papaya, beer, peach. And definitely you would have seen all these flavors which are available in the market in the juice industry. So it reduces the viscosity of these juices. If you see the juice that you get, it's really fine. I mean, it's pure form. It looks like real juice, right? Original juice. So that is done majorly by these enzymes. That magic is done by these enzymes. So they reduces the viscosity of the juice and give it a proper texture. So the third point, if you see, celluloses are utilized for the extraction of phenolic compounds from the grape pomace, and they also reduces the bitterness of the citrus fruit and improve aroma and texture. So these enzymes, they also give the flavor and taste to the juice, and also in citrus fruit, I mean orange, then your mosambi and all these fruits. So it reduces the bitterness from these fruits. So whenever you have a packaged food, whenever you drink a packaged juice, so in that the bitterness is majorly removed with the help of these enzymes, so that it is palatable, so that consumers come again and again to buy those products. So that is why food industry is taking the help of a natural method, that is enzymes, in this case cellulases, right? Yes. So now guys, coming to the next class of enzyme, that is pectinases. So pectinases, again, if you see, these are also majorly used in juice industry, right? So they are obtained from aspergillus and penicillium. So pectinases are majorly obtained from the yeast class of microorganisms. So moving ahead, if we read it in detail, so these substances are found in tomato, right? Pectin is major component of tomato, pineapple, orange, apple, lemon, pulp, orange peel, and other citrus fruit that act as a natural substance for this enzyme. So these are pectic substance. These are, pectinase majorly, this enzyme majorly act on all these food products. Basically, if you see, so these are all majorly citrus products, be it lemon, orange, pineapple, tomato, they all fall in the category of the citrus fruits. So pectinase majorly attack the citrus fruits. If you see the application, it's added to the juice to have a clearer appearance and filterability. So as we read about the cellulases, similarly pectinase is also widely used in juice industry, but majorly in the citrus fruit industry. So if you drink the pineapple juice or the orange juice or the lemon juice from the Tropicana pack, so you will see behind that package, if you see the list of ingredients, I think I should have put few pictures of those packets, so it would have given you better idea. No problem, next time I'll keep it in mind. So whenever we have to discuss about any products that are available in the market, I'll just put a small picture of that product, and also we can look it into ingredients, so you can get a better idea, right? So now coming to the second point, reduces the turbidity and haze generation of the naturally dried fruit juices such as apple, banana, and pectins also improve the color and flavor of the food. So yeah, I mean, we have also read about these. I mean, this is again a repetition. So they give color to the juice. They give flavor to the juice. They reduces the turbidity. They make it very clear. And enzymes, guys, enzymes are 100% safe. These are 100% safe to consume, to eat, because you know the scientists have done such a elaborate work, especially for the food industry. So now we are at a stage where enzymes are widely used and they are very much safe for our health. Why? Because I mean, you imagine you might be having curd every day, right? Especially in South Indian foods, or be it North Indian foods. So North Indian take either buttermilk or curd in their food. Even South Indians, they have buttermilk, or they eat curd rice a lot, right? So I mean, curd is one of the basic example in which microbes are used. So microbes, they release enzymes. So it's a complete chain. I mean, these are 100% safe for us. And if you have any doubt, I mean, definitely you guys would have drank Tropicana at least once in your life. So it was really safe for you, right? So these microbes are, and these enzymes which are used in food industry, these are 100% safe, and they are approved by FSSAI or the other international approving agencies. That is why they are being used in our food, right? Fine. I hope it's clear, guys. So in case you have any doubt, I am always available on WhatsApp. So you just guys can drop me a message over there. And also guys, I'll share with you after once this lecture is over, I'll share a uh, there's a one picture. So it's in table form. So it would give you a clear picture of what we have spoken in a lecture. So what are major enzymes, and in which industry they are used. So I'll do that. I'll share that picture along with you, right? So now coming to the next enzyme, that is glucose oxidase. So again, these are used in juice industry and jam and jellies. So if you see, this is obtained from Aspergillus niger, again a yeast. So glucose oxidase, they belong to a large group of enzyme family called an oxidoreductase. So Aspergillus niger species is widely used for the production of glucose oxidase. So this enzyme is produced by a yeast. Its name is Aspergillus niger. So Penicillium edamatici is widely used fungus for the production, and along with Aspergillus, again Penicillium is also used widely for the production of this glucose oxidase. So if you see here, if you talk about the application, so it has a wide range of application. I mean, this is also used in pharmaceuticals, pharma industry, food industry, and in biofuel. So guys, biofuel is the no talk of the town these days. Even FSSAI is promoting biofuel production. So we'll cover about this when we talk about the FSSAI initiatives. So in that we'll learn how FSSAI is spreading the awareness, amongst all the food restaurants, to not to use oil more than twice or thrice, and to donate that oil which has been used at least thrice to these biofuel companies. So these biofuel companies, they come every day, right, and they collect this oil which has been used, and they use it in the production of biofuels. So we'll talk about this initiative of the FSSAI. And also last year there was a question from this biofuels. I'll also discuss that question with you, right? So that is very, very important. So now coming to the second point, it enhances the flavor, aroma, stability of the food products by removing glucose and oxygen from the diabetic drinks and egg white. So further, again, it's again a common function, you can say, of enzymes. They are majorly, since they are in food industry. So enzymes, they give flavor, aroma, they give the food the stability by removing glucose and oxygen from the diabetic drinks and egg whites. So I mean, all things are common majorly. So I think that is why FSSAI never asked any question from this topic last year, because majority of the functions are same. And enzymes function is basically to break down into simple sugars to give aroma to the flavor, food taste to the food, stability. So that could be the possible reason FSSAI did not ask any question from this topic, right? So now coming to the third point, it improves the color, texture, flavor, shelf life. So I mean, again, it's a repetition. We have talked about this in our previous slides as well. So it improves the texture, color, flavor, shelf life. And fourth point, during food production, it is used for an increased storage life by removing oxygen. So it also removes the oxygen, right? Since it's known as oxidoreductase. So oxidoreductase, basically it removes the oxygen from your food. So if oxygen is removed, the environment is such that if the oxygen is removed, so majority of the microbes cannot survive in that. So it helps in the increasing the storage life or the shelf life of the product, glucose oxidase enzyme, right? So now moving to esterases. So this is again used in beverage industry and cheese industry. So esterases, it's obtained from Bacillus cereus. So if you see, esterases are able to facilitate the splitting of esters into acids and alcohols. So esters are nothing but the long chain of molecules. So these esterases, it breaks down the esters into acids and alcohols. It gives the it breaks down into two compounds, that is acid and alcohol. So what are its applications? So if you see the applications, they play a prominent role in food industry and alcoholic beverage industry, where they are used for modification of oil and fats in various fruit juices to produce fragrance and flavor. So esterase basically since you all are science students, majority of you are science students, right? So esterases, it gives fruity taste, fruity flavor to the food or the drink. So if you see here, it gives the fragrance and flavor. I mean, that fruity flavor, whenever you open that, bottle of Tropicana, or whenever you open that bottle of, be it any juice, real juice or paper boat, there is a fruity flavor to, your juice, or be it your wine, or be it your alcohol. So that fruity taste is because of this enzyme, esterase enzyme. So whenever you drink packaged juice, or wine, or be it your any drink, processed drink. So that you would see that fruity flavor, fruity smell that comes, that is because of esterases enzyme. So in cheese manufacturing, the fruity flavor, yes, that is what I was mentioning here. The fruity flavor are the result of the different methyl and ethyl ester of the short fatty acid chain. So in cheese industry, even if you open the cheese packet, so different type of cheese, they have a different distinct aroma or flavor in them. So that majorly comes